really the main problem in the whole wild world is the drought stress. And we are currently working on drought and insect as well. 2016 World Economic Forum recently just declared that the water crisis is one of the biggest problems in the world. And we are expecting 9.6 billion people in 2050. So then the food security, and then also in order to meet the demands that the population asked for, that we need to really focus on drought stress or water stress as well. So that's our main goal. In order to accomplish or address these questions in our lab, that also like many people are doing it, wheat genome sequencing is critical. Now there is an international wheat genome sequencing consortium that are really focusing on completely sequencing the wheat genome to identify, unlock the whole wheat genome genes and other structure of the wheat genome. And recently, like in a consortium, that International Wild Emmer Wheat Sequencing Consortium, that we were also a part of that consortium, and I'm excited to be a part of that consortium anyways. And recently sequenced the wild emmer wheat, which is just only 2 genomes in there, but it was wild, not elite or cultivated wheats that we sequenced, and annotate the complete genes and genomes, try to understand the evolutionary aspects of that genome, and also domestication, and which was really critical. And all the results that we identified so far are really promising and can be utilized to improve that elite cultivar or bred wheat easily. And we know that from the 15 years ago I started working on wild emmer wheats, it has a big effect on drought, biotic and abiotic stress, and also micronutrient deficiency is critical right now. And we identified that the wild emmer wheat is really high and rich in micronutrients like zinc and iron. And 10 years ago we published a couple of articles that wild emmer wheats were great. So in order to understand the abiotic stress and drought, and or biotic stress like a stem cell phyllite, this is just one example, or pathogen, yellow rust. Some of our group, some of our scientists are working in Israel and some other places as well. UC Davis, University of California, Davis, they are working on yellow rust and other type of rust. It is important to identify, clone the genes, and so that the mechanism and understanding is going to help to improve new varieties in bred wheat as well. So that's what I can say is that first thing that we wanted to understand broadly what the genome structure of wheat is. And so we understand what are the genes available currently in the wheat genome and the wild emmer wheat genome as well. So the genes that we identified and annotated and is now easy to clearly edit or manipulate in order to find drought responsive, insect responsive, or pathogen responsive genes. So that in agriculture, that when we edit the genes, because we know the genome sequences, A, T, G, and starts going there, we can easily edit the genes or knock out the genes so that we will have more water resistant or tolerant or drought resistant, drought stress resistant wheat we can have. So instead of planting a lot or watering the plants or wheat, we're going to maybe water really less, and we're going to use less water. So it has a big impact in agriculture in terms of water usage. This is one example. And then number 2 is that those genome or genes that can identified in wild emmer can easily be applied to bred wheat, which makes high quality bred wheat, for instance, high protein, high micronutrients in bred wheat, which is critical because 20% of our calories proteins are coming from wheat. So everybody is consuming wheat. Wheat is a stable crop all over the world. So increasing the protein content or micronutrient content like zinc and iron in that wheat is important for human health too. So that any sequencing projects will help us a lot in terms of editing, using the CRISPR-9 and gene editing tools to edit the genes, knock out the genes, or overexpress the genes, which if it is really useful to plants and also human beings as well. For instance, zinc is critical in human health. So for brain development and for the kids, and like immune system increasing and immune system ability for kids to immune any diseases is critical. So improving those type of wheat proteins or genes or expression or micronutrient deficiencies will help human and kids as well. That's the main thing. Not only agricultural purposes, but also human health purposes are the main aims when you sequence the genome as well. Because then you'll understand the structure and genes, because then you can play with the genes, edit the genes, manipulate the genes for agricultural purposes and for also human health.